All right, guys, this is part 2 of Mendel and the gene idea, or part 2 of just genetics. We're looking at, starting today with probability. In probability, we've got the chance of an event occurring, and this is usually a number between 0 and 1, 0 being it never happens, 1 being that it always happens. So if you look at flipping a coin, for instance, and you were wanting to know how many times, if I flip this coin, what's the chance of it landing on heads? Well, we know that a coin has 2 sides, so if you flip a coin, you technically have a 50% chance. So in probability, we would say that that is a 0.5 probability. When we're looking at this then in genetics, it gives us the ability to figure out what's the probability of getting an offspring from 2 particular parents, assuming that we know what the genotype is for the parents. So there's 2 rules that govern how we do this. The first is called the rule of multiplication. In the rule of multiplication, let's say we're looking for the probability of getting a big R offspring. Now this could be round versus wrinkled. It could be red versus white, whatever. I'm just throwing out 2 letters here for the example. What's the probability of getting a big R offspring from parents that are big R little r and big R little r? So here's the cross that we've got. Now technically we could draw a Punnett square and see how many boxes out of 4 have big R in them. And that is easy whenever we're looking at just one trait. But this formula that you're looking at will actually be able to be used not only with just one trait, but with multiple traits. And we'll do some examples like that in class. So for this example here, the probability of getting big R from a big R little r and a big R little r parent, well, the big R coming from this parent, technically you would have a 0.5% chance because there's 2 options, big R and little r. So the probability of getting big R from that parent is half. The probability of getting big R from the second parent is also half. When you multiply 1/2 times 1/2, you get 1/4. So the probability is 1/4 or 0.25. And if you did a Punnett square, that would be the same thing you would find in a Punnett square. There's also what we call a rule of addition. In the rule of addition, you use the rule of addition when you're looking for a heterozygous offspring and both parents are heterozygous as well. So now you've got what's the probability of getting big R little r from big R little r parent and a big R little r parent. So when we look at that, what we have to take into account is that the big R could have come from the first parent, which would be a 0.5% or a 1/2 probability, which would force the little r to come from the second parent. That would give you a 1/2 probability. You would multiply that using the rule of multiplication. That would give you a 1/4 probability that the big R came from the first parent, little r came from the second. But you also have to consider the fact that that big R could have come from the second parent and the little r come from the first one. So when you work that out, you get a 1/2 times a 1/2. That gives you also a 1/4 probability. You then add those 2 together to get the probability of getting big R little r is 1/2 or 0.5. And if you worked out a Punnett square, you would see that that is also the case. So a lot of students will take the math involved here and use the math to find the probability of getting certain offspring instead of trying to draw out a Punnett square. Because if you think about in just this one monohybrid cross, that's only 4 boxes in a Punnett square. If you're doing a dihybrid, now you've got 16 boxes. So instead of doing that, you can simply use these formulas to get that. And we will work on some examples in class. The next thing we're going to look at is what I term complex genetics. And these are genetic situations that aren't just normal Mendelian genetics. So it's not just the simple dominant recessive, one dominates the other, you either have it or you don't, that sort of thing.
Sorry, teachers, Common Core PLC meeting. Common Core PLC meeting is in 2203 at this time. Thank you.
All right. So we have incomplete dominance is the first type of complex genetics. This is a heterozygous, is in between the dominant and recessive traits. And these are, of course, looking at the phenotypic expression. So you have a dominant let's say snapdragon flower has a dominant color of red, that would be big R, big R. The recessive expression, little r, is white. But the heterozygote that is formed from these two is pink. And you can see that that is incomplete dominance. One is not completely dominating the other, and you get this in-between version. Now, this is sometimes confused with codominance. In codominance, the phenotype shows both alleles being expressed. So it's not a in-between, but it's a both are expressed. And we see this with blood type. So in blood type, we have an allele A, which gives you the A blood type. You have a B allele, which gives you the B blood type. And you have the little I, which would give you the O. So you actually do have a dominant and recessive being capital I or lowercase I. But you also have an A and a B allele. So you've got multiple alleles there. So when we look at this to find out what an individual may have, you have to take into account that they also have still two alleles. So if they have big I with the A allele and big I with the A allele, or they have big I with A allele and the lowercase I, they have what we call A blood. If they have the B's or the B and the little I, they have what we call B blood. But here is where the codominance takes over. If they have the A allele and the B allele together, we say they have AB blood because they actually express both the A and the B characteristics. Not a mixing of the two, but both are expressed equally. Or they could have the little I, and they would be considered O blood, which would be the homozygous recessive still. So this is a little bit more complex. That's why I term it complex genetics. So you've got incomplete, which is a mixing, codominance, which is both expressed. Another type of complex genetics is pleiotropy. In pleiotropy, one gene has multiple effects. So, for instance, sickle cell anemia, you get blood flow problems, you get fatigue, you get pain, you get brain damage at times. There's multiple effects that are caused just by that simple one gene. So we call that pleiotropy or pleiotropy. Another is epistasis. In epistasis, you get one gene alters the expression of the second gene. So fur in mice is an example of this. A particular type of mice with this case. The big B allele is black coat. The little b allele is brown coat. But there's another gene here that's altering the expression. Big C allele is for pigment. Little c allele is no pigment. So what we find then, if you have a capital C, meaning that pigment will be expressed, then this allele will show you what the color is. So big B, little b would be black because it has the big C. Little b with the big C would be brown because it has pigment and then it has the brown pigment. Down here, notice big B, that should be black except it has little c, which means no pigment. So that mouse is white because it doesn't have the pigment gene. Same here, little c, it should be, or little b should be brown, but because it has little c, it's white once again. So this gene is altering the expression of these genes. That is called epistasis. Next to, the last two of really complex genetics is polygenic. In polygenic, you get an additive effect of two or more genes. And we see this, for instance, in skin pigment. You have, let's say, I'm just putting out letters here to represent this, an A allele, a B allele, and a C allele, and you've got both dominant and recessive versions of all three of those. The more dominant alleles you have, the darker the skin pigment. So if you have capital A, capital B, lowercase c, that's kind of like a middle-of-the-road skin pigment. The more dominant alleles you have, the darker the skin pigment. The more recessive alleles you have, the lighter the skin pigment. And that's called polygenic. The last one are environmental influences. Here you have environment will determine the gene expression. So this mixes people up sometimes, specifically with things like hydrangea, a plant. It has a flower. The flower could be anywhere from pink to purple. Well, that looks like it should be a gene expression. But that's not the case. The flower color is actually influenced by the pH of the soil. So if the pH of the soil is acidic, you get one color flower. If it's basic, you get another color flower. The environment is affecting the expression. The next is the Arctic fox. The fur color is influenced by the temperature. Cold temperatures, it turns white. Warmer temperatures, it turns a nice light brown. And that also is affected by the environment that it lives in the Arctic regions. Another example we could give would be weight in humans, influenced by the diet and exercise that you have, more so than genetics. The genetics is there that also could influence possibly a weight condition, but if the diet is bad and the exercise is bad, then you end up with possible weight issues from that. And that's an environmental influence. The next one here is pedigree. When we're looking at a pedigree, we're going to follow a specific trait over the generations within a family. We do this a lot with humans because we don't like to do genetic testing on humans, obvious in the fact that you got to deal with these offspring. So we don't want to do genetics experiments and then just throw the offspring away, right? There's moral and ethical issues with that. So instead we do a history, a family history or family tree using a pedigree. So let's say, for instance, in a dominant trait, this could be any number of traits, but we look and we have the males are squares, the females are circles. If it's shaded in, it means it has whatever trait we're looking for. In a dominant trait, you can see this one's dominant, but it has recessive kids. So you would say, well, this one, although it's dominant, it's probably a heterozygote. That's the only way for a dominant individual to get a recessive child. So you've got two kids that are obviously dominant, one that's recessive, which does tell you that this one's heterozygote. This individual has a spouse that is dominant, the female spouse here, and they have two kids that are recessive, which would mean this was also a heterozygous, and then one that has that dominant condition. This is carried on one of the autosomes. Another thing we look at with pedigrees is the sex-linked characteristics, which are carried on the X chromosome. This one is sex-linked recessive. One of the easy ways to look at that is that there are more males that have it than females, although in this example, there's not that many. So it would be hard to see that possibly right at the beginning. But you see both parents normal, meaning dad only has one X and his X is normal. Mom has two X's, one of them is normal and masking the other, so we would call her a carrier. Sometimes she's half shaded in. She has a son with the condition, a daughter without it, normal X from dad. All the daughters from him would be recessive, or would not have the condition. So then you see dad with it, mom without it, son without it because he gets a normal Y from dad and normal X from mom. Daughter here with it because he gets infected X from dad. And apparently mom was a carrier. And mom had the other X that also expressed here. So that is sex-linked traits. Carried on the X chromosome. Many of the females in sex-linked traits will be carriers and not express the trait, whereas the males will have it. You see this with color blindness in humans. The last thing we want to look at is inherited disorders. Here we've got two different types of inherited disorders, recessive and dominant. In the recessive, it's only seen in the homozygous recessive individuals. Stuff like cystic fibrosis, sickle cell disease, both of which you see in the U.S. This is more with European descent. This is more with African American descent. But that does not have to stay that way, especially in the U.S. And to have the condition, you have to have both the recessive alleles. In a dominant situation, that could be either the homozygous dominant or the heterozygous individual would show whatever the trait is that you're looking for. Two examples that we see in the U.S. as well, achondroplasia, which is dwarfism, and Huntington's disease, which we call lethal dominant. This one will cause the death of the individual early in life. Usually shows up in their late 30s, or at least in their 30s. Very sad situation because they seem normal up to that point, and then it has a deterioration of nerve functioning, eventually killing the individual. These individuals a lot of times has already passed those traits down to their offspring before they have their offspring. It's a very sad situation there, and it's called lethal dominant because the dominant trait causes the death of the individual.